Welcome back, folks. This particular video lecture is going to talk about protein structure. The next video lecture will talk about protein function. The agenda is quite universal. We have a lot of things to talk about. So we'll do a quick recap on what we know about anabolic and catabolic systems and how proteins fit into that landscape. And then we'll talk about the roles of proteins within living systems. Then our true analysis of protein structure will begin with the building blocks of proteins, known as amino acids, and we'll discuss them in the context of their properties, which are very important. We'll next look at peptides and what peptides are and how they're constructed. And we'll finish off that conversation by looking at how peptides are folded into their final shapes and the role played by chaperone systems within cells. Then we'll take a small tangent and look at diseases caused by proteins of a particular type, prions. Then we take a true journey from very fundamental protein structure called the primary structure, and then we'll make our way systematically through higher and higher levels of protein folding and interaction until we get to the quaternary level. Then finally, we'll finish off by looking at protein domains, what they are. Sometimes these are confusing for students, so I'll try to make it as simple as possible. And then a related topic is structured and unstructured regions. So a quick video on that will show you how that works. And then finally, we'll talk about self-assembly, and that will help us get a good understanding of protein structure. So let's begin by looking at a quick recap of anabolic and catabolic systems. So we told you earlier in a different video lecture that a cell receives food, and these are heterotrophic cells. Plant cells or photosynthetic cells will make their own food. Regardless, the food molecules enter the cell, and once they're inside the cell, they can normally follow two general pathways. One is anabolic, where they build up more complicated structures. And the other is catabolic, where the food molecules are broken down, generally for the production of energy. Both of these are pathways. So there's not a single step that can lead from food to an anabolic structure, such as a DNA molecule. And likewise, the breakdown of food isn't done in a single step. That would be too detrimental to the cell. The amount of energy released in that case would burn the cell to death. So nature has come up with pathways that are broken into discrete steps, and each step converts one substrate into a product. Every step that we know of is catalyzed by a protein, at least in 99% of cases. There are a few exceptions about RNA acting as a catalyst, but we'll save that until another video lecture. But for now, let's keep the general theme alive that biological catalysts are proteins, and sometimes they partner up with minerals and vitamins that are also necessary in our diets. But the general theme is this: without the assistance of these biological catalysts, life as we know it would not be feasible. Somebody's calculated that it would take 100,000 years for a single molecule of glucose to break down in a typical cell under natural conditions. That's a bit surprising, but that's what the science tells us. So proteins are very important catalysts, and every step of a biochemical pathway has a protein controlling its activity. Let's move on to specify some roles of proteins within living systems. Students have no problem listing a couple of these, but any more than that, they tend to struggle. So let's start with the most obvious from the previous slide. So proteins function as enzymes, accelerating chemical reactions. The second most obvious answer is that proteins have a structural region. And the structural reason would be, for instance, producing the cytoskeleton of a cell, or the hair or nails of an organism. The third most common use of proteins is in terms of transport. And the great one that we always use is hemoglobin for carrying oxygen and carbon dioxide. But within the body, there are many more transport proteins. Next, we have motor proteins. Not a favorite of students, but think about it. Your muscles, the muscles of animals, they can generate a force by allowing protein subunits to crawl across each other. And we'll talk more about them in a physiology type setting. But motor proteins are very important, right? Beyond that, we have storage proteins. So these are proteins that can be used to store some type of chemistry for the benefit of the cell. And examples of that, of course, will be calmodulin. Calmodulin can store by intercalating with calcium. So it stores calcium within the cell. And beyond that, we have many other examples of other types of molecules being sequestered, like iron. Iron can be sequestered within a cell in proteins known as ferritins. So those are the obscure examples, right? Receptor proteins. These are proteins that receive information. So the classic one are your visual pigments, your rhodopsins and your opsins. They can partner up with some types of pigments in your eye to capture light and then translate that light into some type of chemical signal. So these are receptor proteins. And at the cellular level, cells perceive the world around them by using receptor proteins embedded either in the plasma membrane or within the cytoplasm of a cell. So talking about gene regulation, we have transcription factors. And these transcription factors are gene regulators. By binding to genes and their switches, they can regulate the degree to which genes are activated or not. Next, we have signal proteins. So signal proteins will be proteins which relay information either within a cell or within the body of a multicellular organism. So examples of this will be your hormones. So your hormones can signal to other cells to change their functionality. And we'll talk more about those in the future. After this, we have all kinds of other proteins that are specialized, maybe for a particular type of organism or a particular type of cell within that organism. And these are evolutionarily adapted. So there could be hundreds of these that are either discovered or waiting to be discovered. So let me just give you an example of these special classes. So my favorite are antifreeze proteins. Antifreeze proteins that are found in the circulating blood of fish and other types of creatures who are subject to freezing conditions of the water in which they reside. So fish have proteins which can interact with ice crystals to prevent the ice crystals from growing and therefore killing the cells in that organism. And the example goes on and on and on. But if you can remember maybe 5 or 6 of these, you will be doing a great job. So let's turn our attention now to the building blocks of proteins. It's important that we know that there's 3 terms that we need to be familiar with. The first is amino acid. The second would be peptide. And the third would be protein. So using the terms appropriately is encouraged. From my perspective, I tend to use the protein term only when the finished product is used by the cell. But during the manufacturing process, I will use the words amino acid and peptides to relay that it's not a finished protein yet. So you may use the same terminology, or you may decide to use some other terminology. Regardless, let's begin by looking at what an amino acid is. An amino acid is a simple small molecule that has dual properties, hence the name amino acid. So one side of the molecule has a nitrogen and the other side has a carbon. So let's put a typical amino acid backbone together. So that's the backbone of an amino acid. All amino acids that we're going to talk about in this course, they have this backbone of nitrogen bonded covalently to a carbon, the central carbon, this one here, and then that's bonded to the third carbon. That's bonded to the second carbon. And the second carbon is the one that we call the acidic part of the molecule. The nitrogen makes the ammonia part of the molecule. Now, depending on whether this is in the water environment or not, this could be ionized, this molecule. Regardless, this is an amino acid. One thing missing is that the carbon is always bonded to one hydrogen. That leaves us with the central carbon having one more free covalent bond. And that is always bonded to something below. Now, the something below is what gives us our different flavors of amino acids. In this course, we assume that there's only 20 amino acids to choose from. So if an amino acid is akin to a Lego brick, then we can say that we have 20 different colors of Lego brick to choose from, each representing a different R group. Okay? So the R group down here could be a simple hydrogen, or it could be a methyl group, or it could be two methyl groups, etc., etc., etc. In most cell biology courses, you may not need to learn every single one of the R groups for amino acids. What you may be required to learn are some of their general properties, and we'll see that on the next slide. But as far as an amino acid is concerned, amino acid consists of a central carbon, an amino group, and a carboxyl group, with the central carbon having a side group, and the side group can have various atomic configurations. What we want you to learn next is that the properties of these amino acids are very, very important. And the 20 amino acids, can be divided into two sets of 10. So we could have 10 of the amino acids on one side and another 10 on the other. Quite nicely, the 10 on this side, they interact with water quite well. So they're hydrophilic, right? So that means they're going to be polar. Hydrophilic substances are polar. And the 10 on the other side are going to be non-polar. They unfortunately don't like to mix with water. Now the polar 10 can be further divided into two batches of 5. So 5 of them are ionized to a large degree. That means they're broken up into both acidic and basic amino acids. So there are 2 acidic ones and 3 basic ones. The remaining 5 down here, so these add up to those 5, the remaining 5 out of the 10 are going to be slightly charged, polar in some type of abnormal distribution of charge along their molecule. Okay. And of course, then the 10 non-polar ones are going to have a charge distribution that makes them unable to mix readily with water. And those are the properties that will now govern how these individual amino acids then flavor the polypeptide or the protein to which they are part of. So knowing that is key to understanding protein function. Let's now direct our attention to the word peptides. A peptide is simply more than one amino acid bonded with a partner. So we can take one amino acid. Let's consider just the core atoms. So those are the core atoms of the amino acid, the backbone. And let's add that to a second amino acid. So there we go. And the second one could be any one of the 20, and the first one could be any one of the 20. The cell will decide which combination to attach depending on the information carried by the DNA. So once you bond these together, you will end up with a backbone that looks like this. Now that is called a dipeptide. And the dipeptide consists of one special named covalent bond. There's nothing special about it, except it has a name. And that bond is this bond right there. And that is called a peptide bond. You only find peptide bonds holding amino acids together. So therefore, peptide bonds should only be applied when you're talking about proteins. You'll notice that the nitrogen atom, the free nitrogen atom, is on one side, and it's right here. And then if you look carefully, you'll see that there's a carbon atom on the far side by itself. And this gives orientation to the peptide. So this side is called the N-terminus. And naturally, this side is called the C-terminus. A good way to remember this is that nature makes polypeptides backwards with respect to the alphabet. N comes after C in the alphabet, but with respect to building these polypeptides, it's the other way around. So what would happen next? Inside a cell, the next amino acid will be added to the C-terminus. So we take this molecule as it is with its single peptide bond, and then we add a new amino acid to the end. And now you have another peptide bond right there between the carbon of the existing dipeptide and the newly incoming amino acid. So this will be called a tripeptide. Now, once you get to this stage, you can change the name to polypeptide. Okay, but naturally anything above 7, 8, or 9 amino acids would naturally be called a polypeptide. So there's your association between amino acids, peptide bonds, C-terminus and N-terminus, and the term polypeptide. Just one thing. Where are these amino acids put together inside a cell? The ribosomes have authority to decode a messenger RNA and then to convert that information peptides using amino acids. So this is important universal understanding of how cells work. Okay. The important thing is that the polypeptide, once it's produced by the ribosome, looks like a string without any internal organization. Now, that's not how polypeptides become proteins. For a polypeptide to become a protein, it has to fold correctly. And normally, as it's being produced by the ribosome, as it grows out of the ribosome, and there's enough of it produced, it will tend to take up an automatic shape depending on the conditions within the cell. So let's look at those conditions first. The most important one is temperature. If you change the temperature, then the folding of the polypeptide will be directly impacted. Another physical condition would be the pH. Depending on the pH of the environment in which the polypeptide is being released, it will also interact and interfere with its folding, therefore giving it an alternative shape under different pHs. Then the list goes on. The next thing is the concentration of salt. Salt concentration is very important inside cells. This ionic environment can also impact the folding of that polypeptide. And the list goes on, and we won't go into that. You know, the presence of certain types of ions, etc., metals can impact the cellular environment. Regardless, if we assume that all those conditions are stable, then whenever the same polypeptide is produced by that ribosome or any other ribosome inside the cell, it will always fold automatically into a particular shape. So the protein will start folding into a particular shape as it's being produced from the ribosome. So this is an automatic activity. That's good, because then the cell doesn't have to keep an eye on all the thousands of proteins that it may be producing at any one moment in time. However, some proteins need a special environment away from everything else in which to fold, because that shape may not be automatic in the cellular environment. So what happens in those cases is, as the polypeptide is being produced by a ribosome, the polypeptide is either directly or indirectly placed inside a protein container. That protein container will allow then the polypeptide to fold safely away from the rest of the cellular environment. In some cases, that chaperone, as it's called, the protein chaperone, may have a lid to completely exclude the effects of the environment. This gives a nice safe place for the polypeptide to achieve its final working configuration, its shape, before it's released to go and do its job. And then that chaperone can be reused for the next polypeptide. So chaperones are simply containers, like vases, into which a string can be deposited to allow the string to take up a natural configuration, a shape. And once it achieves that shape, then the string can be ejected and the vase can be reused. So that's what chaperones are. Okay, somebody asked you what a chaperone is. In this context, that's what a chaperone is in cell biology. So you see the word I'm using the most of today is shape. So if the shape of the polypeptide is controlled, then every time you produce that polypeptide under the same conditions, it'll have the same shape. Therefore, you can guarantee that insulin today in your body looks like insulin next week and the week after. And that consistency is very important. So let's take a few seconds to take an aside. We're going to look at prions and what do these disease-forming proteins do. Where should we start? Let's start by looking at diseases caused by these particular types of proteins. In the Pacific Ocean, there are some islands where there's a natural ritual in the past of eating the dead. So eating the dead was a rite that allowed the spirit of and the wisdom of the dead person to be recycled back into the tribe. So that was the ritual. in the 19th and 20th century, evidence became available of some types of neurological diseases where people were going mad. And it mainly affected the men. So the scientists investigated, and they found that even though women and children ate the bodies of the dead, they were only eating muscles and other types of tissue. The most fat-laden parts of the human body are the nerves, the brain and the spinal cord. And the men got to eat those first. They're the ones that had the issues with mental issues, brain decay, right? And when the scientists actually got hold of some of the tissue from some of these patients, they discovered that they had holes in their own brains. So if I was a tribesman and I ate the remains of my dead and I started going mad, then my brain would exhibit telltale signs at the microscopic level of holes in my brain tissue. What was causing those holes was interesting. It turned out to be proteins, just proteins, not cells, not any kind of other pathogen, except proteins. And they labeled those proteins prions. So prions are disease-causing proteins that can exist in the environment. And since then, we found mad cow disease and many other forms of prions that are present in the wild. So what happens in this case? Well, we're going to use a graph to help us, okay? So this over here will be a normal protein. A normal protein found in the nervous tissue. And suppose that this axis here is stability. The stability of the protein's shape, right? I told you shape is very important. So the higher up you are on this graph, the more stable your shape is. So we found that normal proteins in the brain were stable, relatively stable, under normal conditions. But when you found the protein from these scabies people, or scabies animals, that's the name of the disease, scabies, their proteins were much more stable. And that difference in height between the two tells me that these proteins found in the brains of normal people and the same proteins found in the brains of diseased patients have a different energy profile. These ones are obviously more stable than these. So what was happening? Well, it's something to do with contact topology. Let's represent the normal proteins as circles. And let's represent the scabies proteins as some type of square. In the normal brain cell, you would find these proteins doing their regular job, the normal proteins. If you ate the remains of a dead person and that was contaminated with proteins of this type, then those proteins will be then deposited in your nervous tissue. And they will lie side by side next to the normal versions of those proteins. The sheer presence of this protein here next to this protein would cause this protein to achieve a more stable structure. It would send it from here to there. And then this protein would flip to that shape. Then you had a domino effect. This protein would affect that one, this would affect that one. Then this would take, and this one here would take the same shape as the scabies. And then the process would continue until a lot of these proteins had an altered, more stable shape. And in brain cells, that would form a spike. And the spike would continue to grow inside the cell until it ruptured the cell and then killed the cell. And those deaths of cells were responsible for the holes that were seen in the brain tissue of these men from these islands. these are prions. They're very hard to destroy. You can't use bleach, you can't use any other type of heat. You can burn them and they'll still be stable. So far, this particular type of disease is under control since we've discovered what it was. There was an outbreak in the 1980s in the UK and in certain parts of the world. And you may have known about it. It's called mad cow disease. So that's what prions are. So let's move on rapidly and look at protein structure. I won't spend too much time here unless I have to. So the primary sequence of all proteins on this planet is simply the order in which the 20 amino acids are joined together into a string by the ribosomes. So each one of those circles represents a amino acid, and then we have this polypeptide, and it's held together by, of course, peptide bonds. And that is called the primary sequence of any protein. Okay. Now, as I told you earlier, as the ribosome produces this string, it has the opportunity, in most cases, to naturally take up a structure. As it's being produced, and it starts to fold into a structure. And that is called the secondary structure. And the most common secondary structure, and these are local foldings, right? Local, if I spell this right, that may help. Let's just go back here. So as this string is produced, it will take up some local structure. So those local foldings are called secondary structure. And the most common type are alpha helices and a type of beta sheet. Not the only ones, but the most common that we find in many proteins. So secondary structure is the primary structure carrying interactions at the local level. So the side groups of the R groups of these amino acids, remember there can be 20 different types, they will start interacting with each other. So if this one likes water and this one likes water, maybe the two of them will fold over to form a shape. If this one here doesn't like water and this one doesn't like water, maybe they'll get together to form a water exclusion zone. So now you can see the primary sequence carries the information for automatically generating the secondary structure, either alpha helices or beta sheets or some of the other minor examples of secondary structure. So secondary structure is local folding of a polypeptide into individual shapes. So once this particular polypeptide here is finished, it may have an alpha helix or beta sheet at one side, and then it will have something else in between, and then it may form a different structure like that. So you can see a single polypeptide that has a C terminus and an N terminus. And remember, the N terminus is always made first. The N nitrogen comes out first. That is a polypeptide with 1, 2, 3, 4, 5 secondary structures. Inside the cell, based on what we told you about these conditions here, the environmental conditions, the protein will go on to take up a final configuration where all these parts come together to generate a final structure. Could be a blob, globular, or it could be some type of filament that comes together. So the tertiary structure, the third level structure, is the polypeptide having its final shape. And for most proteins, that's where the term polypeptide ends, and they go to work, and they're now known as proteins. So over 60% of the proteins in a typical human cell are of that type. Individual loner proteins that know what to do. But there is still a significant number, about 30%, of proteins that are formed from more than one polypeptide. And that's where the term quaternary comes in. the fourth level. That only applies to these proteins that need partners, right? So this blob here could require two blobs of the same type to connect together somehow magnetically for it to become the protein. Or in some cases, you may require two of those plus two more, like this. So each of those represents a tertiary structure, a polypeptide. Now we call these subunits. So two square subunits, two square polypeptides, plus two circular subunits made by a different gene may be required to come together to give you your final protein. And this is what we have with hemoglobin. Let's turn our attention now and discuss protein domains. Let's assume that we have a polypeptide that has folded into an alpha helix, and that's connected to another alpha helix, and that's connected to a beta sheet. Pretend that that structure here forms a role inside the cell, and that those two alpha helices and that beta sheet are necessary to form a shape that binds to DNA, right? So here's some DNA, and that shape is necessary to bind to the DNA. Now suppose another region of the same polypeptide, so let's continue it, down here has a configuration like this that can bind to something else. Let's say that it is able to bind to other proteins, right? So other proteins will be like this. Okay. So we have a region of the protein back here, this region here, that can bind to DNA. And then we have a region of a protein, the same protein, that can bind to another protein. So each of those regions is still part of the same polypeptide because it's connected through here. But each region is its own domain. This one here we call the DNA binding domain. And this one over here we call the protein binding domain. And if you wanted to, there are examples of proteins that will have the polypeptide continue and make yet another domain that may be functioning as an enzyme. So in this case, this domain over here may have the ability to act as an enzyme. So a single polypeptide, a single protein, when it's finished, will bind to DNA under some circumstances. It could also bind to a protein using its protein binding domain, and then it can use its enzymatic domain to perform a chemical reaction. So domains are functional parts of proteins that can do independent things. So this particular domain, let's change the color once again. This particular domain up here is independent of this domain, which is independent of that domain, even though they all belong to the same polypeptide. You can have another polypeptide come in and make a more grand domain structure. But domains are simply parts of a polypeptide that have a role that has been identified. This is a great time to speak to you about active sites and allosteric regulation. So we're going to take a few moments of your time and indicate to you what an enzyme does under normal conditions. Suppose that this is my finished enzyme. And it's made up of, in most cases, a single polypeptide. So don't worry about the internal structure, but the polypeptide will be, something like this. So it'll be in there somewhere. So that's my finished three-dimensional polypeptide. And it makes this particular shape. Suppose the substrate for this particular enzyme binds into this region. And that's where the manipulations take place during the enzymatic reaction. This is called the active site of the enzyme. Okay, so let's put that in a nice definitive color. So this is the active site. And the active site normally has a topography, a shape that's compatible with the substrate. So the substrate will come in and a chemical manipulation will take place within the active site. And then once the reaction is over, then the product or products will be released. And then the active site will go back to its original configuration and be ready for a subsequent substrate molecule to come in. So an active site is where the chemistry manipulation takes place. But most enzymes also have other places on their surface that can bind other chemistry, or maybe this chemistry. So other sites are known as allosteric. Allo means other, steric means site. So if something else came and bound here, like this triangle, that could be a hormone, and it bound in here, then that could change the shape of the entire three-dimensional topography of the enzyme, causing the enzyme to then change its active site. So this knock-on effect will change the site there. And now this substrate can no longer bind, or bind with less efficiency, right? And that will change the behavior of the enzyme, therefore affecting the chemical reactions that it can perform. That may be a way of controlling the behavior of chemistry within a cell. Maybe that's a neural circuit or a chemical circuit, as it's more likely to be called. So we'll talk about chemical circuits more in the next video when we talk about protein regulation. But the active site and the allosteric site are important. They are normally applied whenever proteins interact with something else. And lastly, I have to get this word somewhere, the word ligand. It's confusing for students, but it shouldn't be, because all it means is something that binds to a protein. So this particular protein here, it can bind to two things. It can bind to the red molecule. So the red molecule is its ligand, but it can also bind to the blue triangle. And the blue triangle is also its ligand. So anything that binds to a protein, whether it be another protein, a DNA, food molecule, a hormone, a lipid, a sugar, you name it, nucleic acid, it doesn't matter. All those things are called that protein's ligands. Okay, let's turn our attention now to talk about structured and unstructured regions. The regions we're referring to are the regions of a polypeptide or of a protein. As we were telling you earlier, the alpha helix is a pretty ordered structure in that the side groups of the different amino acids are interacting with their neighbors, normally using non-covalent bonds. So they could be using hydrogen bonds, they could be using ionic bonds, or they can be using van der Waals forces, hydrophobic, hydrophilic interactions. Any of the non-covalent bonds can normally be used to stabilize the spiral structure of that alpha helix. And likewise, the beta sheet can be stabilized by lots of hydrogen bonds and other types of ionic interactions and van der Waals forces to give that some rigidity. So parts of a polypeptide or protein that are rigid are called structured. Parts that are floppy, that can have at different points in time a different configuration, are called unstructured. So maybe this region here, let's use a different color. Maybe this region here of this polypeptide can be found in alternative configurations during a period of time. So 1 second is there. The next second, maybe, if I go back to the original color, maybe it's down here like this. And the third second is up here. So because it can fluctuate, it has no definitive structure, we call those unstructured regions. Unstructured regions are important because they allow two rigid structures to move around. So it's important that proteins have structure when they need stability, and unstructured regions when they need flexibility. You can imagine an antibody, and antibodies are normally represented like this, Y-shaped molecules, and they have unstructured regions where the arms of the Y originate. That gives the antibody the ability to form this structure, as well as structures like this. So the arms can be flexible. But the arm itself has to have rigidity. So these parts are rigid, and this part here is the only flexible part. So that will be made of unstructured regions, and these will be made of structured regions. Let's conclude this particular video lecture by talking about self-assembly. Self-assembly is where subunits of proteins come together automatically to generate the required shape that the cell needs and expects. For instance, let's say that we produce a globular protein that looks like this inside a cell. And we produce enough of them, and we want to produce a filament. So one of these could automatically combine with a different polypeptide produced by a different gene. So this was produced from gene A, this was produced from gene B. Every time we produce these two and we release them into the environment in which they operate, they will always take up this shape under the same cellular conditions. But what could happen in a cell that was operating at a different temperature? Maybe these proteins could shift their structure, their topology, and generate a different shape, and that's exactly feasible, right? Suppose that at a higher temperature, protein A produced by protein A produced by gene A has a slightly different shape. So instead of producing this at, say, 21 degrees Celsius, at 25 degrees Celsius, the A now has a different structure, like that. So you can see this little knobby thing here has changed and is rearranged to a different side. Now, maybe it can no longer interact with B, because B doesn't fit together with A that well inside the cell. Maybe now it fits together better with C. And now C and A will form a union, and that will lead to a different outcome for the cell. This happens more often than you think. Our cells are very sensitive to pH, salt conditions, and of course temperature. We spend 2/3 of our calories every day trying to maintain our internal environment so that the proteins have a consistency that leads to self-assembly of the same structures every time. But I want to give you one quick example of where this system can be really, really useful. Okay, so let me see if I can pick a different pen color. I'll use purple. So let's explain this now. Suppose we have a cell that's operating at 3 different conditions: 21 degrees, 24 degrees Celsius, and let's call it 27 degrees Celsius. Okay, so under these conditions, the subunits will form with each other just 2 structures. There you go. That's what the subunits can do. They can form this dimer. Okay, that's all they can do. Assume that the same protein is made and released by the ribosome, and this time it's operating at a higher temperature. Let me just use a different pen and a different width. So this time it does this. So the front end is the same, but the back end gets a small indentation. So that means now you can have a lot of these subunits stacking up on top of each other like that. And it keeps going on and on and on. And now you'll have a long filament, where here you had a blob. And maybe the exact same polypeptide at 24 degrees is elevated to 27, and it has a completely different orientation. So now it has a slight curl on it because of the higher temperature, like that. And then the next subunit will come in here, and it will cause a bending, like this, right? My drawing's right. And that will lead to the formation of a spiral. So the same protein at 21 degrees forms a small blob, at 24 degrees forms a filament, and at 27 degrees forms a spiral. Maybe these are necessary to change the behavior of the cell so that at different temperatures, the cell can respond in different ways to protect itself from damage. Or maybe this is a pathogen that decides at 21 degrees not to replicate, but at 27 degrees to replicate and produce baby viruses or baby pathogens. So this is how fascinating the world of proteins can be, all because proteins operate based on their shape and their charge. Thank you so much.